So thank you all for listening to my presentation today on the development of a new method of detecting and quantifying steroid hormones, especially stress hormones, in right whale blow. I'd also like to thank all my various co-authors for their laboratory and drone-related contributions, among other things. I couldn't have done it without any of you. So a brief introduction into right whales, for those of you that don't know. Right whales are long-lived, slow to mature and slow to reproduce, feeding on some of the tiniest animals in the world, copepods. They generally die for around about 20 minutes at a time while feeding. And interestingly enough, even though we know a reasonable amount about their distribution, at any given time, a proportion of the population is just missing. We don't know where they are. They do require good acoustic habitat to support important behaviors, such as cow-calf communications in their critical habitats and elsewhere. So this is their range from sort of the breeding grounds in Georgia and Florida up to Cape Cod Bay and Bay of Fundy, where they've been traditionally feeding. However, there's been a shift since around about 2010, so that the population has become more prevalent in the Gulf of St. Lawrence as opposed to sort of the Bay of Fundy, Cape Cod area. And this has been thought to largely be a result of climate change and a changing distribution of their prey. And now we're seeing around about 40% of the population appear in the Gulf of St. Lawrence every year. Threats to right whales span a range of things. Ship strikes and entanglements can have both lethal and sublethal effects. Climate change, I've already mentioned, changing the quality and distribution of their prey. And we also have noise that we know causes stress in the right whales. And why is this all important? Well, the population has been in decline since 2010, especially since 2015, as they've been showing up in the Gulf of St. Lawrence, so something's going on. But we don't think it's just deaths. Tim Fraser from St. Mary's University did some explorations of how much this population should be reproducing, how much it actually was reproducing, and they're only reproducing around about 30% of their potential. So how much of the decline is related to actual mortality and how much is related to reduction in offspring production, we don't know. However, it's likely to be a result of health or stress related concepts. And that's why we're here. We do know that exposure to human activities are causing changes to the health of right whales and other marine mammals. And that we do know that stress responses have been identified as being key to delineating the full extent of impacts from noise, contaminants, etc., on these animals. This is why stress responses have been identified in many fauna as a priority area of study. Now, the gold standard in many animals, including humans, is the levels of hormones in the blood. However, it's not only possible to get blood from free-swimming whales, and even if you can, the act of chasing them and handling them often leads to changes in those levels, so you're obscuring the baselines you're looking to detect. Alternatives have been explored, ranging from baleen to blow, and each of these have pros and cons for collection, and each represents different timeframes over which the hormones are averaged. Now, blow has been seen to have much promise, as demonstrated by Burgess et al. It's relatively low impact and can be collected systematically, unlike feces, which is more opportunistic. It's believed to reflect similar short timescales to blood, which does mean it has the potential to include chase capture effects. It also is a little tricky because you don't get a known amount of the substrate on the petri dish or whatever you're collecting with. So you need some sort of way to normalize between one sample and another. And you often get low amounts collected, which can be challenging for quantification, especially with assay kits. So the objectives of this work was to explore a new method for quantifying stress hormones that reduced the threshold of detection and increase the level of blow collected to improve the proportion of samples that we collect from the field that are useful. We also wanted to look at ways to eliminate the close approaches by vessels to the whales to reduce that chance of influencing the sample levels. In terms of lowering the thresholds, the gold standard there is mass spectrometry, which is capable of detecting multiple hormones in a single sample at lower levels than assay kits, even though it costs a little more. So we partnered with Vancouver Aquarium to get a lot of blow from their Pacific white-sided dolphin Helen to support the method development and validation. The resulting method for analyzing the samples was that they were thawed from -80 degrees where they had been kept since collection in the field. And then the samples were extracted using ethanol on an agitation plate to make sure we got everything off the Petri dish. Those fractions were then transferred to glass tubes and subjected to a solid phase extraction process. The resulting extracts were then reconstituted in 0.1% formic acid for the LC mass spec analysis on a Q-trap mass spec. Now, this was involving 10 microliters of each sample being objective at time 0 on the graph, and then subjected to a gradient method of mass spec. And by that, I just mean that the carrier solvent starts at formic acid and switches over gradually to methanol, then flips back to formic acid. And this is designed to separate out the hormones and give you a higher sensitivity at the back end. On the maximizing the sample size, we looked at the Heligai drone that was pioneered by Vanessa Perotta and her colleagues in Australia. And we were in the process of obtaining one of these that they used in this case for collecting blow from humpback whales to look at bacterial content when the company was bought by a larger conglomerate, and this product was no longer available for sale, so it vanished off the market. I then approached Gab Cocker, one of Canada's top drone racers, about trying to replicate that sort of drone. And he, with a PhD in physics, saw the opportunity to take his skills and apply it to science. And so he was quite excited to work with us to generate the Pequod. He was unfortunately unable to join us in the field to test out his new piece of kit. So very smiley Liam Alders here was sent along in his place. And this is a first-person view drone. So he would sit in a chair and fly it from the Coriolis, which we were in the field. And the Coriolis itself is a sort of mid-sized research vessel capable of a science complement of around about 12 people. This is the results of that effort, and I'll take you through the field process a little here. Firstly, we would detect the right whale. We'd see it on the horizon. We'd move over. We'd try and get pictures of the whale from over the top for our photogrammetry. At this point, Liam would then take off for his 3 to 5 minute flight, approach the whale and try and collect the blow, as you see here, on the Petri dish so that we would have our sample. He would try and go through the blow as many times as he possibly could to maximize the amount of material on the dish. And so the fact that he had a very versatile, very agile racing drone was critical to achieving this. So did it all work? Well, we did get cortisol, which was our main target, but we also got 2 other hormones concurrent with the extract process that was targeted specifically at cortisol. And they were corticosterone and progesterone. We also got urea using the assay kits. And we did get 22 field samples in 2019, including our full complement of 20 North Atlantic right whales in our permit. The results we saw were similar in many ways to that seen by Burgess, which was encouraging. And we have here the North Atlantic right whales on the left and the other animals in our process to date on the right there. The progesterone, we got the same sort of absolute levels as Burgess et al. Did, but after we normalized using urea, our levels were 10-fold lower. So we need to look into exactly why that happened here. However, the distribution across the different animals and the variability looks realistic. And we have some other things that give us a little extra confidence in our results, which I'll come back to a little later. Corticosterone was not previously explored. However, the pattern here and the extent of the variability again seems to match that of the cortisol and other stress hormone. So this gives us further confidence that what we're seeing is real. Like Burgess, we explored the effect of age, and you can see that the variability and the levels were higher in that sort of central ages rather than the very young and the older animals, which is similar to the trend seen previously. We looked at the number of blows, and we got differing trends here, none of which were very big. And we'd like to look at this further, but we need a bigger statistical sample size to do that. So we did get 100% quantification for our samples for these 3 hormones, which is a step above the 86% previously seen. And we do have that potential to expand the extraction process like adding acetone into the mix to try and expand that to other samples. A couple of case studies to give us extra confidence. We accidentally sampled Ruffian twice within around about 40 minutes, and that was largely due to Liam's gung-ho nature, because this was the second and third samples he's taken. And he didn't really wait for confirming that the animal was a new animal. So we had been basically following Ruffian on the Coriolis for an extra period of time. And you can see here that the cortisol and corticosterone have gone up a little bit in that second sample, but the progesterone has stayed about the same. So it looks like we might be seeing a little tiny bit of the chase effect here. We didn't get any pregnant females, which would have given us a good indication if the progesterone samples was working. However, we did get 3 females within the range. Unfortunately, none of them were having calves the next year. We did get 1 female that was lactating at the time because she was present with her calf, and she has low progesterone, which is consistent with lactation. So this was encouraging as well. So back to our objectives. Did we develop a new methodology that was improving the proportion of samples that were useful? Yes, we did. We got up to that 100% mark. We also eliminated the need to approach the vessels closely, and that may be why some of our levels are slightly lower than previously seen. So we're quite happy that we achieved the goals we set out. What are the next steps for this work? Well, obviously, we need to do a full statistical analysis looking at various factors, but before we can do this, we need the IDs of the animals, which are only preliminary right now, to be confirmed by experts with the New England Aquarium. We also want to analyze the 2020 samples that we have in hand and explore other extraction solvents and procedures to try and expand that hormone suite to get other things like testosterone and a few others, so that we can get a broader picture of the health of the animal. Critically, we really want to collect more field samples, because it's only with a weight of samples, it's only with having a lot of samples that we can begin to tease apart the potential impacts of different human activities on the stress and thus health of the animals, much like environmental change, like climate, that will all be operating in unison. So if you want to start teasing out the actual impact of any given one activity on the animals, we're going to need a larger sample set to do that. So thank you all very much for listening. It takes an absolute village to make this work, and I want to thank everyone at DFO, at Vancouver Aquarium and the Coriolis and elsewhere that contributed to this work. We really couldn't have done it without you. And I will be available for questions. Thank you.